It's important to note in meiosis the cell divides twice. So we have two divisions, meiosis 1 and meiosis 2. Meiosis 1 is basically actually the reduction division. Yeah, where the cell divides and forms the first step. Meiosis 1, it divides to form two daughter cells which are haploid. Yes, so meiosis 1 consists of a mother cell dividing to form two haploid cells. That is, if humans, the cell was 46 chromosomes, the product of the two daughter cells after meiosis 1 have 23 chromosomes each. Then meiosis 2 is actually just replication of mitosis. It's just mitosis. So 23. Each dot cell divides to give two more dot cells. So in total we have 4 dot cells. You can see here. So each daughter cell produces each 2 daughter cells, identical to each cell. So we have 4 daughter cells which are haploid. So let's talk a little about meiosis 1. It's still the same. Meiosis, after interphase, everything that happens in interphase, multiplication of the chromosomes and G2. Now during prophase, there is the same things as mitosis. There is the same process as mitosis, except that now there is synapsis. There is a process called synapsis in meiosis 1. Synapsis is basically the pairing of these homologous chromosomes. See that they're pairing up. That's synapsis. There's pairing of homologous chromosomes. So there is every other thing. There's disintegration of the nuclear membrane. There is formation of spindle fibers, and there is synapsis, pairing of homologous chromosomes, and there is also crossing over. There is crossing over. Then metaphase 1. During metaphase 1, homologous chromosomes align to the center. Remember, I'm giving a difference here. During mitosis, we said sister chromatids sister chromatids align at the center. Here, the homologous chromosomes align at the center during metaphase 1. And then during anaphase 1, homologous chromosomes separate. So what is separating is not the chromatids at the kinetochore. The chromatids are not separate. No, a full chromosome with a sister chromatid is going to the pole. So it is a homologous chromosome is separating from its fellow homolog. Yeah, basically. So if we had, for example, our sex cells, let's assume we had a male. So during prophase 1, they are going to synapse together. Oh, is that a verb? Okay, synapses will occur, and the X and Y are going to come together. They're going to pair up. During metaphase 1, X and Y align at the equator, and during synapsis and during anaphase 1, the Y chromosome goes on one pole and the X chromosome goes on one pole with its sister chromatids. That's a major difference. And then telophase 1, the same thing happens as in mitosis, which is formation of the nuclear membrane, decondensation of the chromosome. Not yet. It doesn't yet decondense. Sorry about that. It doesn't yet decondense. So after telophase 1, the cell moves directly without any passaggio. There is no passaggio into. There is no intermittent, if I miss it. Sorry. Oh, it's like I'm forming new words today. Anyways, there is no intermittent. There is no interface between telophase 1 and prophase 2. Yes. Immediately after telophase 1 and cytokinesis, of course, there is prophase 2. Prophase 2, remember, I said is immediately is just mitosis. So exactly everything that happens in mitosis happens in prophase 2, in meiosis 2. So we'll not dwell on that. Okay. Genetic variation. So there is genetic variation. We said the daughter cells in meiosis are not identical to the parent cells. They are not identical to the parent cells, contrary to mitosis. Now we're going to see why. The first thing is crossing over. There is crossing over which happens in prophase 1 and independent assortment in metaphase 1. We'll explain all these terms. Crossing over happens in prophase 1. Remember, these are in prophase 1 we have synapsis, which is the pairing of the homologous chromosomes, and we can see here that they are sister chromatids of homologous chromosomes exchange genetic material. You see where they are crossing here, and you see this little orange has captured the little blue area, and the blue has captured the little orange area. That is crossing over. So they're kind of exchanging their genetic material. Yeah, and that's basically crossing over after synapsis. Then we have independent assortment, where homologous chromosomes which are aligned at the equator can separate during anaphase in two different ways. Here we have where homologous chromosomes on the left actually depends on the formation of the spindle fibers. Yeah, because the spindle fibers are the ones pulling them apart. So this, here we have two blues that have moved on one side and two reds that have moved on one side. Now, and it can happen in another way where blue and red move on one side. You can see here, and the other blue and red move on one side. So these are two different possibilities which give genetic variation in meiosis. So basically meiosis occurs because sexual reproduction in animal cells, in eukaryotic cells, I beg your pardon, sexual reproduction. And so gametes, that's why you are basically different from your parents, because of the process of meiosis. So thanks to meiosis, you're not. So random assortment can give 2 to the 23 different possibilities. And here's the figure, 8,388,608 different combinations of gametes. Basically, difference between mitosis and meiosis. Mitosis, we said the daughter cells are identical to the parent cells. Here, in meiosis, they're not identical. Mitosis daughter cells are diploid, 2N. And meiosis, they are haploid, 1N. And all the little subtle differences we gave during prophase, anaphase, and metaphase, like the crossing overs and the synapses and all of that. Okay. So I think we've done a pretty decent job. I hope it's really well understood. If not, I'm available in the comment section. Basically, the comment section is really available for any worries you have. And how we draw and label chromosomes? Well, I think I've really explained this already. Chromosome is the DNA exists as one long coil. Yeah, we can really see it's uncondensed here. That's chromatin during interphase, and during mitosis we have condensation. And remember, our chromosomes have to replicate during the S phase and form 2 sister chromatids. And these sister chromatids are joined at a point at the center called the centromere.